Nitrogen is used by life forms to carry out many of the functions of life. This element is especially important to plant life. Yet nitrogen in its gaseous form is almost entirely unusable to life forms. It must first be converted or fixed into a more usable form. The process of converting the nitrogen is called fixation. After nitrogen is fixed, it can be absorbed and used by plants and subsequently by animals. The process of nitrogen being fixed, used by plants and animals, and later returned to the atmosphere is referred to as the nitrogen cycle. After the nitrogen is ingested by the bunny, it becomes part of the bunny's DNA. Before a cell can divide, the DNA must replicate. DNA replication is the process that a cell uses to copy its chromosomes. It takes place in the nucleus before the cell divides. The chromosomes are made up of DNA, and the DNA contains the instructions for protein synthesis. DNA replication begins with one double strand of DNA. The DNA molecule unzips to form two single strands, and the DNA zips back up. The first phase of mitosis is prophase. A cell gets the idea that it's time to divide, so first it has to get everything ready. Next is metaphase, where all the pieces are aligning themselves for the big split. The DNA lines up along a central axis, and the centrioles send out specialized tubules that connect to the DNA. In anaphase, the separation begins. Half of the chromosomes are pulled to one side of the cell, half go the other way. And then telophase, now the division is finishing up. This is when the cell membrane closes in and splits the cell into two pieces. Finally, for interphase, this is the normal state of a cell, and we suppose that when it comes to cell division, you could call this a resting state. It's just living its normal life, getting ready for another. Prophase 1, chromosomes replicate, homologous chromosomes are paired together, and in metaphase 1, chromosomes align at the center of the cell. Anaphase 1, mitotic spindles pull homologous pairs to opposite poles, and in telophase 1, mitotic spindles disappear and cell division begins. In prophase 2, the DNA that remains in the cell begins to condense and form short chromosomes. Each chromosome pair is a centromere. In metaphase 2, all of the chromosomes line up along the center of the cell again, and in anaphase 2, we see that the chromosomes split and move to opposite sides of the cell. They don't divide up the DNA between the new cells, they split the DNA that exists, and each daughter cell will get one half of the DNA needed to make a functioning cell. Telophase 2 shows the DNA completely pulled to the sides and the cell membrane begins to pinch. When it's all over, you are left with 4 haploid cells that are called gametes. The eventual purpose of the gametes will be to find other gametes with which they can combine. When they do, they will form a new organism. Finally, protein synthesis takes place. A portion of the DNA molecule unwinds, exposing the gene responsible for the specific protein. Nucleotides, with the help of an enzyme, move along one side of the exposed gene and form messenger RNA. Messenger RNA binds with the ribosome so that the mRNA can be decoded by the ribosome. The message is read 3 codons at a time. Each codon stands for a specific amino acid. When the codon is read, the corresponding amino acid is activated by an enzyme. A transfer RNA molecule has 2 ends, a binding site for a particular amino acid and an end that has a particular sequence of 3 nucleotides. Transfer RNA molecule attaches to and carries the activated amino acid to the ribosome. This is continued with the other anticodons until the entire message has been read. Then all the amino acids are brought in a sequence, forming a polypeptide chain and a protein. You can see that there has been a mutation in the chain. The baby bunny has a mutation where one of its paws is shorter than the other. Because of this, it dies and the nitrogen is released back into the atmosphere, completing the nitrogen cycle.